These are the first set of notes for your Chapter 12 cell division, cell cycle, mitosis, meiosis notes. So we're going to go ahead and start by reviewing basic vocabulary and really just review from ninth grade so that we're all on the same page and we can start talking about this without having to back up and go over everything over again. So I'm going to go ahead and draw some chromosomes. We're going to start with an organism and it's going to have 6 chromosomes in it. Sorry, I'm moving my little clock around so you might see that. And I'm going to try and alternate colors. So I have a green and a blue chromosome, so it's 2 chromosomes. I'm going to make this chromosome shorter, and we'll explain why in a second. I would encourage you to pick 2 different colors as well. They don't have to be green and blue, but they should be 2 different colors. And the reason that these are 2 different colors is because one set is from mom and the other set is from dad. So maybe mom is the green chromosome, so all the green chromosomes come from mom, and all of the blue chromosomes come from dad. But here is an organism with 6 chromosomes. Humans have 46 chromosomes, so this is a hypothetical organism that only has 6, and there are organisms that only have 6 chromosomes. And 6 chromosomes. Sorry, it's a little pixelated. I'm in Paint right now just because I don't have any other way of doing this. So 6 chromosomes or 3 pairs, right? And this is also why we drew some pair, like this first pair was longer to differentiate it from the second shorter pair. And then I even added some squigglies here to differentiate the third pair from the other 2. So this is pair 1, that's pair 2, and that is pair 3. All right, these 2 chromosomes, the first 2 on the left, are what are called homologous. And homo just means same. So homologous chromosomes are chromosomes that contain the same information. They carry the same genes, which is just a unit of information. So we're going to say copies of chromosomes that contain... Actually, you know what? I think I can type this and it'll be a little easier to read. I can. Contain the same type of info. So basically what we're saying here is that if mom, come on, where's my, there it is. If mom's chromosome has, let's say, an eye color gene right here on the top, I'm going to box it in. That means dad's chromosome, his equivalent chromosome, has an eye color gene at the exact same spot. And anywhere along these chromosomes, whether it's mom's version of the chromosome or dad's version of the chromosome, they all contain some version of the same gene. And that's going to be true for any pair of chromosomes. So they contain the same info on homologous chromosomes. If you have an organism in this case that has 6 chromosomes or 3 pairs of homologous chromosomes, we call this number diploid. So I'm going to go ahead and write that. And diploid means that you have 2 members of a pair. You have 2 chromosomes in the pair. The entire pair is present. And mathematically, we simplify this and we say that diploid is 2n, basically you have both chromosomes are there, for a total of 6 chromosomes. So 2n equals 6 would describe this set of chromosomes for this organism. Right. Excuse the siren for 1 second. I don't know if you can hear that. All right, if we only had 1 chromosome in the pair, we would say that that is haploid. So haploid means that you only have 1 member of the pair. And mathematically, we would say instead of 2n, we would just divide everything in half because you have half the number of chromosomes now. So we would just say n equals 3. All right. And we'll work on that more. The only other thing I want to go over here, I'm going to scroll down a little bit. Let's say that these chromosomes in a cell are going to get replicated. We're going to duplicate the chromosomes. We're going to copy the amount of chromosomes. So what is that going to look like? So here we go. We're going to make a copy of these because the vocabulary does change a little. So this was pair number 1. And when the chromosomes copy, the entire chromosome gets copied and attaches the copy at a point, right? They connect to each other. So same thing for the other chromosome. The chromosomes are going to copy and they're going to connect at a point. I'm going to make a little dot here for the connection. And the point at which the copy connects to the original is called the centromere. The point at which the copy connects to the original is called the centromere. And I'm only going to show you the first pair, but obviously all of the chromosomes in an organism would do this same process. So you would have the same thing going on for number 2 and the same thing going on for number 3. Oh, let's do it. Let's do it. Go ahead. Go do it with me. So here's pair number 2. Remember, pair number 2 had the shorter chromosome. Pair number 3 had the squiggly chromosome. And remember, the copy and the original are connected at what is called the centromere. Whoops, there's an E on the end of centromere, by the way. Right there. Right. Once you have the DNA copied, you might say, wow, there's twice as much DNA, there must be twice as many chromosomes. And at this point, it's tempting to think that, but the amount of chromosomes, the number of chromosomes actually stays the same. So the number, let me type this out, the number of chromosomes stays the same, but the amount of DNA doubles. And this is a little bit tricky. The number of chromosomes is equal to the number of centromeres. And although I didn't draw the centromeres up here on the top, even these single-stranded chromosomes, even before they made a copy of themselves, they still had a centromere. I just didn't draw it up here, but those centromeres are present. Right, so the number of chromosomes is constant in this case, but the amount of DNA is what doubles. So don't let that kind of fool you. And I don't know why my typing moved up, but it did. So the other vocab we need to talk about is once the chromosomes have made a copy of themselves, the copies, each individual copy, we don't call them the original and the copy anymore, we call them sister chromatids. We call them sister chromatids. And sister chromatids are identical. Because one is a copy of the other, they have the same genes, right? They have the same information on them. So sister chromatids are copies of each other. If you want to compare, let's say I want to compare this chromosome from pair number 1 to this chromosome from pair number 2. Well, they're not copies of each other, so they would be called, you might remember this from honors bio or regular bio, these would be called non-sister chromatids. Non-sister chromatids. All right, so this is the basic vocab that you're going to need to know for this unit, and I'm going to go ahead and switch over to the big sheet. We're going to head down to this figure 5 down here in the bottom left. All right, so what we're showing is we're showing a diploid cell. This is a really, really simple cell. There's only 2 chromosomes in this cell, and I want you to go ahead and color code these. So you can pick whatever colors you want. I'm going to pick, let's stick with how we've been doing this. We've been doing this green and blue. So here is green, and just so that I don't have to do this over and over and over again, I'm going to go ahead and color everything green all at once. So here's the green chromosome here. Here's the green chromosome there. And you can go ahead and do this with me, and I'll walk through why it's kind of working out the way it is. I just don't want to switch between the different colors in this version because it gets very, very cumbersome. I'll do blue for my other chromosome. All right, so let's talk about what's happening here and why the cells are dividing the way they are. So here's your diploid cell, and because you have 2 chromosomes in here, the diploid formula is going to be 2n equals 2. I'm going to write in red. Oops, I didn't mean to do that. So 2n equals 2, right? Both pairs of both chromosomes in this pair are present, so this is a diploid cell with 2 chromosomes. So 2n equals 2. And remember, one of these is from mom and one's from dad, so I think from memory we did green was mom and blue was dad. I think. If not, you can go ahead and switch that. And if you want, you can add the M and the D from mom and dad all the way through. Otherwise, the color coding is fine. And remember that once DNA makes a copy of itself, once you've replicated your DNA, the number of chromosomes is still 2 because you have 2 connection points. You have 2 centromeres still. Remember, the individual single-stranded chromosomes have centromeres also. But the individual parts, remember the copies are called sister chromatids. Sister chromatids. And then once the cell starts to go through mitosis, and we'll see the steps of this in a second, the chromosomes line up single file in the middle of the cell. And this is absolutely critical because this is going to be a little bit different depending on if we're doing meiosis or mitosis. So I'm going to go ahead and say that the chromosomes line up in the middle, single file. Single file. So this organism only has the 2 chromosomes, but in humans, since we have 23 pair or 46 total chromosomes, you would have an entire line, right? Actually all 46 chromosomes would line up all in the middle of the cell. They'd all kind of get squished together. So here it's only 2, but every single cell, all of the chromosomes line up single file. Don't let the orientation of this, if you could rotate this or if you tilt your head to the right, don't let the orientation of the cell throw you off. Sometimes this might look like a horizontal line of cells, sometimes it might look like a vertical line of cells, but the point is they're all lined up in the center of the cell. The chromosomes line up in the center of the cell. Cannot emphasize that enough. The individual chromosomes are what line up. These structures here, if this is an animal cell, the ones that I've kind of made those little red dots, you should recognize these as the centrioles. Plants have a similar structure. They're not centrioles, but they also have a similar structure. And all of these little fibers are spindle fibers. Remember that spindle fibers are just, I'm going to type this one. Spindle fibers are made of microtubules. tubulin is the name of the protein, and this is just cytoskeleton. Remember, this is a type of cytoskeleton. I'm writing this where I have space, but you guys might want to write this a little bit somewhere else. And remember that this requires assembly, right? You have to link up different proteins, different tubulins to make this cytoskeleton. This is an anabolic process that requires ATP. If we want to write this, we can say, again, I'm running out of room, but you might want to write this somewhere else. Let's say assembly required, need ATP. And then after the chromosomes divide and mitosis is finished, the cytoplasm will split in two, and you have two diploid cells that are daughters. They're called daughter cells. And I'll bring the M and the D down here to the end. Oops, M was green. All right, M was green, D was blue. And these two chromosomes, remember, they are homologous to each other. These are homologous chromosomes that have the same info. They have the same traits. So same info / traits, and they are identical to the parent cell. They're identical to this initial parent cell. They're smaller because they're half the size. This parent cell split in half, but they're identical. So the basic formula is you are going to copy the DNA. The chromosomes are going to line up. Right? They're going to line up in the middle of the cell, and then the cell is going to divide. That's kind of the overarching process here. Copy, line up, divide. All right, let's go right up to this diagram here. So this is the cell cycle, right? Cells are constantly dividing and doing their job, and dividing and doing their job, and dividing and doing their job. And most of the cells in your body are going through this cycle and are somewhere in this cycle at any given point in time, right? Really quick though, let's talk about why cells would even divide in the first place. So kind of up here on the top, we can make a little note. Cells are going to divide in order to, the first thing is to grow, right? You have to grow an organism. You started as a single cell, and then you built a trillion plus, I mean, not just 1 trillion, trillions and trillions and trillions of cells make a body. You are trillions of cells, so you're obviously growing. And the second thing is to repair. I'll say repair or replace damaged cells. Right? If I cut myself, those cells are not permanently missing. I don't have a permanent gash in my arm. The skin cells will undergo mitosis and kind of close the gap and heal me, right? So cells, yes, they grow, they divide to grow, but they also divide to repair or replace damaged tissue. All right. So we're going to start in this cycle. We're going to start right after cytokinesis, and we have to add, because my little my lines that separate these all kind of got invisible. Add these lines in here. We're going to split this up. So this is kind of like one section up here. Here's a section. So we got 4 sections here, and we're going to start over here in G1. So right after cytokinesis, right after the parent cell, the original cell has divided, we're starting with small cells, right? Because the cell is split in half, so the cell is half the size. The daughter cells are half the size as the parent cell, right? So these cells are small, and the first thing on their agenda is to grow, right? So G1, it stands for gap 1, but you can also think of it as growth 1. And the primary function is just to get bigger. You have to increase your volume, you have to increase your surface area. This is going to entail making more cell membrane. It's going to entail making more cell structures. You're going to have to make phospholipids. The smoothie R is going to be busy making those. You're going to have to attach phosphate groups to your fatty acids. You're going to have to build any integral proteins that are in the membrane, any channels, any pumps. aquaporins, anything that's part of that membrane, cholesterol has to get synthesized, and that's just in the cell membrane itself, right? So the first thing a cell has to do is to get bigger. So we're going to increase in size. Also, remember, mitochondria and chloroplasts are kind of independent of your cells. Remember, they are symbiotic prokaryote. They're not prokaryotes, but they're related to prokaryotes. They're derived from them. They've evolved from them. So we'll say symbiotic prokaryotes. Remember, they divide on their own. They have their own DNA. In our cells. Binary fission is actually the way bacteria divide, so that's one more evidence that they are descendants of bacteria. So these are all the things that are happening in growth 1. So let's make some more notes. Oops, let's say we're going to build phospholipids, proteins for membrane. Let's make a note that this is an anabolic process, right? Anytime you're constructing something, you're putting things together, you're synthesizing, you're doing dehydration synthesis. This is an anabolic process that needs ATP. So we can go ahead and add that. And while we're at it, because the cell that we're using here is the same cell that we just worked with, if you want to pause the video and take one quick second and go through and color code all of these chromosomes, go ahead and do that. Remember, green and blue are the colors I used, but you can pick whatever you want. I'm just going to do this really quick. Should have done this first. My apologies, I forgot. All right. And these were diploid. Right, so these were 2n equals 2 diploid cells. All right, let's move on to S. So S is the second phase here. S stands for synthesis. This is where your DNA gets duplicated. So what you're going to see when you go from G1 to S is that your chromosomes have twice the amount of DNA, right? You don't double the number of chromosomes because the number of centromeres stays constant, but you do double the amount of DNA, right? So S, I'll write the word out, S stands for synthesis. Let's, on the side, make a little note. We'll say that you still have 2 chromosomes. 2 chromosomes, 2n equals 2. That's our formula. So twice the amount of DNA, right? And just to remember, let's take this blue chromosome here. That each part of this double-stranded chromosome is called a sister chromatid. And they are copies of each other. They have the same genes. They are identical. Sister chromatids. All right, next phase, gap 2 or growth 2, if you want to think of that. You've replicated DNA. You have to check it. So there is going to be a bunch of enzymes that check, basically, did you match the A's and the A's and the T's and the T's when you copied this? When you copied the DNA, did you copy all of the strand or did a chunk of the strand get left off? You're going to check this for errors. Right, and any repairs that need to be made are going to be made during G2, right? These enzymes that are responsible for checking and repairing DNA are inherited from your parents because DNA codes for proteins, and these proteins are, in this case, enzymes, right? So you inherit the genetic code for these enzymes from your parents. And one of the things that, this is kind of luck, I mean, it's not kind of luck, it is luck, is that, hope that you got a good pair. I'm going to actually say that. Hope you have a good copy of these repairing enzymes from mom and dad, because the better they are at finding errors, the less likely you are to actually get cancer. And we're going to talk about the role of cancer here in a second too. These proofreading enzymes are incredibly important. So I'll write that over here. Proofreading enzymes, check for mistakes. And those mistakes are called mutations, right? The more mutations you accumulate over time, the more likely you are for the cells to be damaged, potentially to get cancer. So you want a good copy of these proofreading enzymes from your parents. So after you've done G2, everything has been checked off, you're good to go. The cell will continue through mitosis. Remember that mitosis has your different stages. So I'm going to draw little lines here. You've got prophase, where the chromosomes condense, the nucleus, the nuclear membrane dissolves. You've got metaphase, where the chromosomes line up in the middle of the cell. You've got anaphase, where the sister chromatids get pulled apart. And then you've got telophase, where the cell membrane starts to pinch in, and the cytoplasm also starts to pinch in, right? So mitosis is division of the nucleus. It's division of the nuclear genetic material. Cytokinesis, which follows immediately after that, is when the cytoplasm starts to pinch, right? And you can tell that there right in the name. Cyto is right there in the name. Right, and this is all kind of one right after the other, and this process is very, very quick. Cells don't spend a lot of time dividing. They spend most of their time in everything else. So at the end of mitosis and cytokinesis, your product, we can go ahead and write this too. In this particular cell, we have two members of a homologous pair, the blue one and the green one, the two chromosomes. And remember, one was from mom and one was from dad. I think green was from mom, one from mom, one from dad. And remember, in humans, you have a lot more. You have 2n equals 46 chromosomes. But in this particular one that we used in this diagram, there's only 2n equals 2. Right. Before I forget, G1, S, and G2 are all referred to as interphase. So cell division includes mitosis and cytokinesis, and that's, again, pretty quick. The vast majority of the time a cell spends in is interphase, right? All the way around interphase. This last little component here, G0, is for some cells, not all of them. Actually, most cells don't really go into G0, but some cells exit the cell cycle, and once they kind of are mature, that's it. They don't divide. And these are cells that you don't get second chances of. When they're dead, they're dead. So these cells include muscle cells, some nerve cells. We've actually learned that some nerve cells can regenerate, but most of them don't. Liver cells do this. However, liver cells can repair. If you like cut a portion of your liver off, maybe you're donating it, and or whatever, you have a surgery where you have to get some of your liver cells, or you literally cut a chunk of your liver off. What am I talking about here? The cells adjacent to your missing liver can detect that. There are actually mechanisms for them to signal each other and to say, hey, we're missing a whole chunk of liver. And in that case, they can actually repair. But for the most part, your liver cells are there, and that's kind of it, unless there's been some damage to them. right? So big picture, this is the cell cycle. Let me zoom out. And we'll walk through how this is regulated next. So off to the right, you have one more diagram that we're going to talk about. Oh, before we do this diagram, let's talk about cancer really quick. Remember we just said interphase is where most of the cell spends its time, or the cell spends most of its time. So in a normal cell, we can do this right in between these two diagrams. A normal cell spends about 90% of its time doing its job. And by doing its job, we mean making whatever protein, like let's just say you're a pancreatic cell, right? In that case, your job is making insulin or making glucagon, right? Let's say you're a muscle cell. Your job is to synthesize a lot of ATP for contractions, right? Let's synthesize, let's talk about a liver cell. In that case, your job is to detoxify and, well, perhaps to make a lot of lipids. So in G0, in S, and G2, when we say the cell is doing its job, it's existing and doing its thing, that's what we're talking about. That's where most of the cell spends its time, right? And this is necessary for you to be healthy and to live. If a cell is dividing, it can't do its job, right? It's by definition not doing its job. Dividing is an all hands on deck endeavor. So normally, 10%-ish of the time cells are going through mitosis. If you accrue mutations and that impacts how the cell divides, and you have a cancer cell, these numbers are essentially flipped. 10% of the time you're doing your job, you're in interphase, and 90% of the time you're dividing. Right? And this is bad for two reasons. The first is that if you're dividing so much, you're not doing your job. Right? So the cell just by default isn't working at making insulin or glucagon or whatever it's supposed to do because it's dividing too much. The second thing is that because when cancer cells divide and they're pushing up against other cells, they disrupt their adjacent cells' ability to do their jobs. And this is part of the reason why when you have cancer you get so sick. Part of it's because the cells themselves are not working anymore because they're dividing too much, but they're also putting pressure on adjacent cells, and that contributes to all of the symptoms that different cancers have. But let's talk about how this is regulated, what checkpoints there are in this cycle. So this is our last diagram. Notice that there are 3 checkpoints. You have one after G2, one during one during metaphase, during mitosis, and there's another one immediately after G1 before synthesis. So let's look at this G1 checkpoint. It's at the end of G1. This is right before the cell starts to copy its DNA. And the main criteria is that the cell is big enough, right? The cell has gained enough cytoplasm, made enough cell membrane in G1. The cell size is normal. If that's a go, you can go ahead and progress, assuming the rest is still okay. So growth factors, these are usually hormones, not always, from adjacent cells. Say hormones, say slash signals from other cells that tell you that you can divide. And also, if there's too many cells close to you, the physical contact of those cells can basically say, hey, there's no room. So adjacent cells, if there's too many of them, or there's too much pressure against the cell, like actual physical pressure, adjacent cells can basically say, hey, I'm in your way, you can't divide. That's called contact inhibition. If you were curious, contact inhibition. But basically, these two rely on you having space to grow. If all of those are met, check, 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 you progress on to S and you copy your DNA. There's no break or check after synthesis. You go right into G2. At the end of G2, there's a check to make sure, though, that your DNA replication is complete, right? You've adequately or accurately copied all of your A's, T's, G's, and C's. They're all, the sister chromatids are connected at the centromere. You're not missing chunks of chromosome on either end or in the middle. The cell size is still adequate, right? If all those things are a go, check, 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 then the cell will progress into mitosis. In metaphase, there's one more check. This is called the M spindle checkpoint. And basically, the cells check to make sure all the spindle fibers are attached. Remember, here's your chromosomes, your double-stranded chromosomes. Each line's a sister chromatid. They're attached at the centromere. Right? The cell makes sure that these spindle fibers are attached to the centromere. If it is, and it is in all the cases, however many chromosomes you have, everything's good to go. Check. You can proceed and continue to divide. Right. So those are the 3 checkpoints. If a cell fails any of those checkpoints, there is a system in place to prevent the cell from dividing, and this is called apoptosis. Apoptosis. This is pre-programmed cell death. So the cell ends its life. The cell basically says, I'm not good enough. I didn't connect my spindle fibers to my centromeres. I didn't adequately damage or replace or repair damaged DNA. And remember, cells have an opportunity to redo this. So they have, the proofreading is, it's like a second chance. They have like a reassessment, if you will. But if they still don't fix the problem after that, then they go through apoptosis, right? Maybe the cell is not big enough after G2, and it's not fixed for whatever reason. It doesn't get rectified. The cell will undergo apoptosis. So this is pre-programmed cell death. It's planned, and the amount of apoptosis that happens in your body is like billions per hour. I'm making that number up. I'd actually have to do some research on how much it is, but it is quite a lot of cells per minute do apoptosis. It is normal for cells to fail the cell checkpoint somewhere in your body, and it is normal for them to do apoptosis when they can't fix the problem. What isn't normal is when cells don't care if they didn't meet a checkpoint. Think of the checkpoint like a stoplight and a cell just running through the stoplight in like a speeding car or something. That's what happens when you get cancer. Cells really could care less at this point if they have errors or mistakes. They don't care about the checkpoints, so to speak. They continue to divide. They divide, they divide, they divide, they divide, they divide. And because they have all of these unfixed mistakes, these mutations, what you get are cells that accrue thousands and thousands and thousands of mutations and mistakes that never ever stop dividing. That's what cancer is, right? Batches, just clumps, tumors are just massive cells that never learned how to stop dividing because they didn't recognize these checkpoints. They didn't honor the fact that they were supposed to do apoptosis, right? So that's kind of what happens when you have cancer. It's misregulation, mismanagement of the cell cycle, right? So we will continue with the big sheet next time. I hope these notes helped, and I will see you next time.